Bismillah ar-Rahman Rahim. We start another paper four. Your paper four is very near. It's on Tuesday, coming Tuesday, and it's Saturday today. So we just have a few days to prepare for that. Now, in this, I'm going to discuss two or three planning questions. You know, the 6 marks questions, which everybody must know how to handle those questions. Now, if you look at this diagram, this is a simple water bath. Simple water bath means this is actually pathetic. Why is it pathetic? Because you have a beaker. You have a beaker in which you have water, you have a conical flask, and you have a thermometer. And the biggest problem is this Bunsen burner. Now, if you have to maintain the temperature at say 35 degrees Celsius, what are you going to do? You're going to leave the burner on and then you're going to wait. You're going to look at this thermometer and you say, okay, now it's 35. So you switch this off. But then the temperature drops down to 32. Then what do? Then again you switch it on. So this is not a very good water bath. It's, as I said, it's pathetic. But we use this all the time in our labs, and we expect you to come up with this that this is not very accurate. This is here we say the word accurate. It's not very accurate because actually when the temperature went up to 35, and if this room is very cold, then slowly this temperature is going to fall. So there is no way to maintain the temperature. And then of course you look at the temperature. You can't be looking at the temperature all the time. So the temperature falls and then again you switch the Bunsen burner on and then you wait for it to go up to 35 and then again you switch it off and then again it cools down. So this story is going to continue. So this is not a very happy story. So what we use is a thermostatically controlled water bath. These are, of course, this is the word that I want you to use. Thermostatically controlled water bath. Now, these of course run on temperature, run on electricity, and these of course will thermostatically control. They have an on-off switch, just like you when you're baking some brownies in the oven and you fix the temperature at a certain level. You say, okay, 120 degrees or 200 degrees when you want to bake something, and then it maintains that temperature. It will switch on and off. So it is done electrically. It has some sort of a thermostat in it, just like you switch on your boilers and the water heats up and then it switches off. Same thing with the ACs. You put the AC at a certain temperature, the AC will switch on and off. So this is called a thermostatically controlled water bath. Now, this is the word that you must use. If you just say water bath, and of course, some brilliant student also writes bubble bath. And when I ask them, why did you write that? They say, Miss, oh, I meant water bath. So, I mean, the jokes apart. If you wrote bubble bath, I'm not going to give you a mark because just you decided to have a bubble bath. So you have to be very clear about either just don't say using a water bath. You use this terminology using a thermostatically controlled water bath. That is going to get you a mark. If you just say water bath, you might not get a mark. Now, the planning question which I have in mind is this one. It's again a 6 marks question. So there's 6 marks which you have to get 6 out of 6 in this. Photographic film consists of a plastic sheet coated in crystals. Read the question carefully. The crystals are fixed to the plastic sheet by gelatin, which is made of protein. If the gelatin is digested by protease enzymes, the crystals fall off and the film will become transparent as shown in figure 1.4. So before the protease treatment and after the protease treatment, what has happened is that the enzyme is going to digest the crystals and the crystals will fall off and the film will become transparent. Just read the question carefully and keep this in mind when you are answering the question. This is the important thing, is that you must keep this in mind. Now it says plan an investigation to determine the effect of temperature on the activity of a protease enzyme using photographic film. So now what is the independent variable? Independent means I, which I have changed. Independent variable is always that which I have changed. Now what have I changed? I'm going to change the temperature. So you're going to give me 2 points about that. So at least 2 different temperatures. And then how do you maintain those temperatures? Using a thermostatically controlled water bath. using a thermostatically controlled water bath. So you've talked about the independent variable. Now let's talk about the dependent variable. Dependent variable, I've told you in the previous videos as well, is what are you going to measure? What are your result? So it says, what was it saying in the question? The crystals are fixed to gelatin. If the gelatin is digested by protease, the crystals fall off and the film becomes transparent. So what are we measuring? We're going to measure the time for the film to become transparent. Will it take 1 minute? Will it take 2 minutes? Will it take 10 minutes? Or are we going to be talking about in seconds? Okay, it took 30 seconds, it took 40 seconds, it took 1 minute, it took 1.5 minutes. So what are we measuring? We are measuring the time for the film to become transparent. Or you can say till the film becomes clear, because after it said in the question itself, it will become transparent. You could have used the word clear. Or you could have even said for the crystals to fall off, because it gave in the question the crystals fall off. So how much time does it take for the crystals to fall off? 10 seconds, 20 seconds, 30 seconds. So what are you measuring? You're measuring the time for the film to become transparent. So 2 marks for independent, 1 mark for the independent variables. You got your 3 marks. Now what are the other 3 marks? What else are you going to control? Now, here comes a very important question. Now, do you see these two equations which I've written? Proteins, protease gives amino acids. So if there are going to be 100 amino acids or 200 amino acids, what's going to happen to the pH? The pH is going to fall. Similarly, in the question on, if ever you get a question on protease or lipase, remember the products are acids. In protein, they are amino acids. In fats, they are fatty acids. So what are fatty acids going to do? What are amino acids going to do? They're going to lower the pH. It's going to become more acidic. Now, how do you maintain the pH? You use a buffer. That's part of the syllabus point which I talked to you in the first video. So you use a buffer to maintain the pH, because if there are more and more amino acids are going to be made, it's going to lower the pH and the enzyme will get denatured. The enzyme won't work. So use of a buffer to maintain the pH. Then shaking it, well, of course you have to give me some point about the shaking the thing. Then you have to talk about the constant variables. What do you mean the constant variable? What should you keep constant? The concentration of the enzyme or the volume of the enzyme. I always say write both volume and concentration of the protease. Then another point which you can write in nearly in every experiment. Repeat the experiment 3 times and calculate the mean. So you repeat the experiment at the different temperatures and then you calculate the mean. So if you decided to take temperature 30 degrees Celsius and 35 degrees Celsius, then you do 3 readings on 30 and then you do 3 readings on 35 and then you calculate the mean, so that if there is any anomalous reading, anomalous means if there doesn't fit the trend. If there is an anomalous reading, then you can remove that and do it again, because you don't add the anomalous reading in the when you're calculating the mean. And then you talk of the safety precaution. On one safety precaution is what? It's a protease. Protease is a protein digesting enzyme. If this falls on your skin has living cells in it. And those proteins will be digested, so it can damage your skin. So use gloves when you're handling the protease solution. So this is a safety precaution. So remember this, that you have to get your full marks in this part of the question. Now I will read the exam report to you of this part of the question, part E. There were many detailed and carefully planned investigations, with some candidates gaining maximum marks. Almost all candidates described a suitable range of temperatures. However, very few were able to describe in sufficient detail how these temperatures would be maintained. It is important when describing how to control temperature that it is qualified by using a thermostatically controlled water bath. A water bath alone does not keep the temperature constant. Many candidates were able to identify the key variables that should be kept constant and described their experiments being repeated at least 3 times at each temperature. A few candidates included a prediction of the result, but this is not necessary in a plan. So I do not want you to give me a prediction that, okay, this is my prediction, this is what is going to happen. We don't want you to give us a prediction. We didn't ask for a prediction. And some students really think that is the important thing in giving us a prediction. We don't want you to give us a prediction. I know many times I know which students are going to get an A, which are going to get a B and a C, but I don't tell them because I know maybe in the last month they try very hard and the one I'm expecting a C might get a B. So I'm not going to predict what is going to be the outcome of this experiment. They haven't asked you this. They've just asked you to plan. So now I'm going to give this to you in writing, at least 3 temperatures, 30, 35, 40. And you write the units, degrees Celsius, use a thermostatically controlled water bath, measure the time for the film to become transparent, use a buffer solution to maintain pH, shaking the film after applying the enzyme, volume and concentration of the protease should be kept constant, repeat 3 times at each temperature, so that means we will do 9 times, and use gloves when handling the enzyme solution. So very simple, many marks for it, write a lot so that you get your 6 out of 6. Now, coming to another planning question. This is a little difficult, so I'm going to take my time in explaining it to you all. It says dialysis tubing acts as a partially permeable membrane. We don't use the word semi-permeable anymore. So, dialysis tubing acts as a partially permeable membrane and can be used to represent a model cell to investigate osmosis. Plan an investigation to find out how different concentrations of sugar solutions affect the movement of water into or out of the dialysis tubing. Now, as you haven't read the past question, so I don't expect you to really be knowing that, but I'll just show you the diagram which was in the previous part of the question. So this was given to you in the previous part of the question, that there was an elastic band holding the open end of the dialysis tubing, and there was a dialysis tubing in place, and then there was a large test tube which had something in it. But this was the first part of the question. Now I'm going on to the planning question. Now, what is a dialysis tubing? Dialysis tubing is a type of partially permeable semi we don't use anymore, so it's not a correct word to use. Tubing used in the separation of molecules from the blood during dialysis. Now you can see how you fill it up and then you close it and you tie it up, and then and then of course you see how it changes over time. Now in this diagram, can you see what has happened? So the water has moved out and you can see this volume has gone up. So if water has moved out, then this you can either measure the volume inside here, or you can measure the volume here which has increased. So volume here will decrease and volume here will increase. So you can either do that or you can do that. So in this experiment, now it says dialysing tubing acts as a partially permeable membrane. Plan an investigation, different concentrations of sugar solution. So you are going to be changing that. So that is the independent variable. So take at least 3 concentrations of sugar. 5% sucrose, 4% sucrose, and 3% sucrose. Anything you want to say. So at least take 3 concentrations of sugar. Then what are you going to measure? You're going to measure the volume of the dialysis tubing or the volume of the fluid in the large test tube. That's up to you. Either you measure the mass and the volume before and after of the dialysis tubing, or you measure the mass or the volume of the dialysis tubing or the test tube, whichever you want to measure. So this is the test tube. And in this you have, this is the dialysis tubing, which you have tied a knot here and a tied a knot here. This is acting as a model cell. So either water is, in this we have the sugar solution. So water should either move in or water should either move out. This is what you've got to figure out. So it says plan an investigation to find out how different concentrations of sugar affect the movement of water into or out of the dialysis tubing. As I told you before, no predictions are needed. Just tell me how to do the experiment. So when you put in the 5%, did water move in? Did it move in more or did it move in less? When you put in the 4% sugar solution, so if you put in a 4% here, what happened? Then if you put in a 3%, what happened? So this is how you've got to be planning an investigation. Don't give me the results. Don't give me the prediction. Okay, this is going to happen. This is going to happen. No, I don't want you to tell me that. Now, independent variable, as I said, was at least three concentrations of sugar. Dependent variable is measuring the mass of the volume before and after. So the first thing we talk about is the independent variable, which is what you decided to change, is three concentrations of sugar solution. You can even say four or five, that's fine, but it should be a minimum of two is what is required. I always say, okay, talk about three or five on different variables. So independent variable should be three or five. So I've said three concentrations of sugar solution, 4, 5, and 6%. Then you measure the volume or the mass before you immerse that dialysis tubing into it and after you've left it in it for about a minute or so. So measure the volume and mass of the either the dialysis tubing or the contents of the test tube. So this is the test tube and you've got the dialysis tubing in it, right? So either you measure the mass of this before and after, or you measure the volume of this fluid here before and after. So please understand this, what I'm trying to say. Now, the other variables that you need to control, and you can only get 3 marks for this, even if you give me all this, which I've written in red. Volume of water in the test tube, volume of sugar solution, temperature of this, where you place this, then type of the dialysis tubing, and then type of the sugar. Have you used glucose? Have you used sucrose? This is not that one you make 4% sucrose, and then you make 5% glucose. Sucrose is a disaccharide, glucose is a monosaccharide. So the type of sugar must remain the same as well. So out of any of these points, the maximum I can give you is 3 marks. Then a little bit about how you're going to maintain the temperature. So you maintain the temperature using a thermostatically controlled bath. Now, if you wrote that here, it's fine. You can write that here and you can write it again as in the explanation as well. Then make a model cell. You knot the tube at both ends. Then you measure the mass using a balance or you measure the volume using a measuring cylinder. So you have to give me the name of the apparatus which you're using. And then the last point is repeat the experiment 3 times in total so that you can calculate the average and you can remove any anomalous readings. You know, anomalous means those which are do not follow a trend. So this was the second planning experiment. Now coming to a third planning experiment, starch in apple juice makes the juice appear cloudy. The enzyme amylase can be used to remove the starch. This makes the apple juice clear. So originally the apple juice contains starch, which has these sort of it makes it cloudy. Cloudy means it's not very clear. But when you add the enzyme amylase, that's going to digest the starch to maltose, and maltose then dissolves in the fluid. So it makes a clear juice. Amylase catalyzes the breakdown of starch. Plan an investigation to determine the optimum temperature for amylase activity in a sample of apple juice. Best temperature. So what is the independent variable? Is the temperature. So you take 3 temperatures. Then what is the dependent variable? What are you going to measure? The cloudiness of the juice, or how long does it take from the cloudiness to become clear? So that would be, so temperature would be the independent variable, and the fact that how long it takes it to become clear would be the dependent variable. Then how do you do it? So you maintain the temperature using a thermostatically controlled water bath. Then you sample it after a set time, like after every 30 seconds or 1 minute. Then the iodine solution test, you can easily take a sample of it and add iodine to it and see if all the starch has disappeared. Then you note the time taken for the starch to disappear. Then you maintain the pH using a buffer solution. Then volume of the apple juice should be kept the same. The concentration of the apple juice must be kept the same. The type of the apple juice must be kept the same. The enzyme concentration, how much amylase did you add to it? It's amylase in the form of a solution. So was it a 1% amylase solution? Was it a 2% amylase solution? So the enzyme concentration and the volume of the enzyme. So if you said add 2 ml 1% amylase solution, so you got 2 marks for that. Then how much is the volume of the iodine solution that you added? And then of course repeat the experiment at least in a total of 3 times, and then come out with some safety. When you're using the enzyme, wear gloves, or when you're cutting up the apples, then you are careful that the scalpel is away from you. So the independent variable would be at least 3 temperatures. This is the one that we're talking about. And you write down the temperatures. Then the dependent variable would be the time taken for the cloudiness to disappear, or you can say the time taken to digest all the starch, because you're using an enzyme called amylase. So using a thermostatically controlled water bath to maintain the temperature would get you another mark. Then take samplings every 30 seconds. You see, you can't say take a sample after 3 minutes or 5 minutes, because it might have all the starch might have been digested in 1 minute, and you are saying take a sample after 3 minutes. So taking samples every 30 seconds, which is humanly possible, and then test with iodine solution, and then volume and concentration of the apple juice should have been kept constant. Then the type of the apple juice, was it Shazan, was it some other make, country or something. Then volume and concentration of the enzyme solution. Whenever you're talking of a liquid, remember to talk about the volume and the concentration. And then the volume of the iodine solution. How much iodine solution did you use? Or if you're going to test for Benedict's, then you'd use the how much are you going to use the Benedict's. Then another thing which you can write in every experiment is repeat 3 times in total, so that you can then find the average and you can see what is the trend. Then the safety, you can also write that in the end. In every practical is use gloves when handling the enzyme, or when you're cutting up the apple to take out the apple juice, then you be careful and keep the scalpel away from you. So a total of 6 marks, and I'm sure everybody could have got the 6 out of 6. If you wrote more, because some are only, we can give you 2 marks for that. At times we can only give you 3 marks for this. Now in this case, I can only give you three even if you wrote the whole thing. So these are the ways that you handle the planning and questions. These are the 6 marks planning questions, and I hope this is now helpful to you. We've done 3 questions, and this should be a nice revision for you how to handle these questions. Thank you once again. Thank you very much, and thank you for watching. Thank you for the comments. Thank you for all the thank yous, and wishing you all the very best.